Hi everyone. In this video we're going to be looking at a slide of human lungs. Now lungs are always very difficult to prepare because most of the lung is just empty space really, so that's why what you're seeing here is very compressed. Now what we're going to do is, for the most part, we're going to try to look for bronchi and bronchioles, and at the end we're going to try to look at a few of the better preserved alveoli. Okay? So we'll start by just scanning around, and one of the first things I see just at the edge of this section is what looks like a bronchus. Now what you're seeing here is the lumen. The epithelium is right here. And the reason I think it's a bronchus is because right here there's a piece of cartilage, and there's another piece of cartilage right here. This is one of the intrapulmonary bronchi, so it's not going to be completely surrounded by plates of cartilage. What you have is just a few pieces of cartilage that are kind of disconnected from one another, and those are found in the tunica adventitia. So everything to the inside of this is going to be the tunica media, tunica submucosa, and the tunica mucosa, which will include the epithelium. Now we're going to look at that at higher magnification in a moment. I want to just scan across. We have a couple of blood vessels here. These are pulmonary vessels, pulmonary artery and vein. And then over here, we see a couple of examples again of the bronchiole tree. So this one again is a bronchus. It's not complete again because it was cut off. This is the lumen over here. And the reason I think this is a bronchus is because right here is a gland, a seromucous gland. We'll take a look at that one in high magnification in a moment as well. Okay, so again, a relatively large lumen, and we can still see some seromucous glands in the tunica submucosa. In this case, we don't really see, as far as I can tell, at least at this magnification, we don't see any cartilage, but the presence of these seromucous glands tells us that we're still looking at a bronchus. Whereas when you look at this structure here, we're not seeing any evidence of any kind of glands here, and we're not seeing any evidence of any cartilage. So here we're looking at a bronchiole. Okay, so let's take a look at each of these at a higher magnification. So let's slide over to the larger of these three structures, or the largest of these three structures, this bronchus here. And let's switch over to 100x magnification. And so here, this magnification, you can clearly see, again, we have some cartilage here. There's another piece of cartilage right here. And this staining seems to show there's a bit of calcification going on in this case here. Again, so there's a piece of cartilage, so we're looking at the tunica adventitia here. I am seeing a little bit of smooth muscle here, so right there, try to get that. No, so the pointer's not going to get any sharper right now. But right there at the tip of that pointer, the fuzzy pointer, is kind of a more eosinophilic area that is smooth muscle. So this is going to be the muscularis mucosa. So in this region is where we would have the submucosa. Anything to the right of this, in this case, would be the lamina propria. Again, we can kind of see that here as well. There's a little bit of fragments of muscularis mucosa visible here and here as well, and here. You can see that it is fairly discontinuous still. So what we're still seeing is that spiral of muscle going around the outside of this lumen. It's not a continuous layer of smooth muscle in cross-section yet, so the smooth muscle is not a very prominent component of this structure yet, but we're starting to see it become a bit more prominent, much more so than we would have seen in a trachea or one of the extrapulmonary bronchi. So everything in this area here would be the lamina propria, and on this side here, what we have is the tunica submucosa, and I suspect what we're going to see here is probably glandular tissue. So that's probably one of those seromucous glands. So we're going to still see those in the tunica submucosa as well. Let's go up to 400x and confirm that. Okay. So yes, what I'm seeing here is a little bit of mucus cells and some serous cells here. So this is a seromucus gland. There is another one over here as well. So again, mucus cells, they look very empty. Serous cells don't look as empty. Okay. And so again, what we have is a seromucus gland found in the submucosa. So that's what we're looking at, submucosa here. There's a blood vessel in here. Blood vessel? No, that looks a bit more like maybe muscle, maybe connective tissue. And so we're seeing here again that cartilage. We're seeing a couple of chondrocytes here occupying a single lacuna. There's another isogenic group right here. Right, this is being a little sensitive. Okay, so two nuclei very close to one another, so another isogenic group. Okay, so we're clearly seeing some cell division going on here. Again, this difference in staining here is likely to be the effect of the result of some calcification. So there's a bit of damage going on to this cartilage here. Okay, so again, here's that muscularis mucosa. And so in this region here is the lamina propria. This region here would be the tunica submucosa. And again, what we're seeing here is still a relatively pseudostratified columnar epithelium with, on the surface, we have some cilia. Okay, so again, this would be a bronchus. It's an intrapulmonary bronchus, but still relatively large, so one of the larger ones. Let's zoom back out a little bit so I can find the other one more easily. Okay, let's slide over. Where was it? There it is. Okay, and again, right here, we can see, again, an epithelium. And right here we have another one of those seromucus glands. Let's zoom in on that one. So go back to 400x. And so again, right here you can see some of these mucus portions. We see these empty looking cells. They look very much like goblet cells. The nuclei are pushed off to the bottom. The rest of the cell is filled with mucus, which does not get preserved very well during typical slide preparation, which is why they look empty. And then a lot of the other cells, or a lot of the other parts of this gland, we have cells that do have cytoplasm fairly clearly visible. Again, relatively basal nuclei. Let me just slide over here. So you can see the nuclei are kind of at the edges, the outer edges of this gland, and the cytoplasm is a bit more filled in. One of the characteristic features of mucus versus serous glands is that mucus glands tend to have a larger lumen, a wider lumen, whereas serous glands tend to have a very small lumen, so it's very difficult to even detect in this slide especially. And again, here we have what looks like still a pseudostratified epithelium. Again, you can kind of make out that there are some surface specializations still, so we've got some cilia there. Right here is lamina propria. We have some small blood vessels here, some capillaries, and probably venules. Right there is a bit of smooth muscle. Okay, so this is that muscularis mucosa. And everything beyond this, that's the submucosa, which will contain the glands. Okay, again, in this case here, I'm not seeing any cartilage specifically, but that just might be because it's out of the plane of section, or maybe it's just not visible in this region. It might be just off to one side where it was cut off. Okay, but it's still a bronchus just because I can see, or would be classified as a bronchus because I can still see some of these seromucus glands in the submucosa. Let me zoom out again and try to find that bronchiole so we can see the difference. So here is one of the bronchioles. Okay, let me zoom in on this one. So go back to 400x. And here. Again, we have an epithelium that seems to still have some surface specializations here. We're starting to see a lot more nuclei down here in the tunica mucosa in the lamina propria. A lot of those will belong to lymphocytes. We have a little bit of smooth muscle visible here, so we have a muscularis mucosa, and then there's some connective tissue beyond this. We don't really see much within that connective tissue though, so I don't see any glands here. We can scan around the outside of this whole structure, and I don't see any glands, and there's no evidence of any cartilage anywhere in here. It's a relatively thin wall, so I would have to classify this as a bronchus. Now this space that you're seeing here is just an artifact of preparation, so don't think that that's anything in particular that is characteristic of a bronchus. It's just, you know, the cells that are separating from the basement membrane in this case here. Just a bit of damage during slide preparation. Okay, so what we have here is a bronchiole. And again, we can see a bit of smooth muscle here. So this is the muscularis mucosa. So this is the lamina propria. And down here will be the submucosa. And beyond that, we have some alveoli. Okay, so there's not much else in terms of the structure here. So again, there's no evidence of any glands, and there's no evidence of any cartilage, so we would have to classify this structure here as a bronchiole. Okay, now this bronchiole is likely to be starting to show a transition of the epithelium that's going to be a bit more simple columnar as opposed to pseudostratified. It's a little bit difficult because of how tightly packed these nuclei are to really show this. Let me try to maybe zoom in on some of these regions, see if we can get a little bit more detail. Let's go to 1000x. Increase the contrast a bit. Again, I'm not using any oil here, just because I don't have that handy. But you can still see that there's stuff on the surface. In this case here, it may not actually be cilia anymore. You might be seeing a little bit of this eosinophilic material, might be cell protein. So many of these cells are actually producing cell protein, which is kind of like a non-stick coating for this epithelium, so that when it does collapse, these two epithelia do not get stuck to one another. So it's kind of like the Teflon of your body. You kind of have this material that prevents these two membranes, these two epithelia, from sticking to one another when this structure collapses under negative pressure. Okay, so let me zoom out a little bit, maybe go out to 100x so I can see a bit more. And let's scan around a little bit more. Again, we can see some pulmonary blood vessels, so these are clearly arteries that we're seeing here, nice and round. Okay, here is another component of this bronchial tree. It's not very round. It's kind of difficult to tell from here, but we could be looking at a respiratory bronchiole. I don't see any sites of gas exchange though, so it's probably more likely to be damage. Here again we have a couple of structures. So we have an artery, we have an accompanying vein, and we have another, what looks like to be a bronchiole. Again, right now I'm just looking at based on size, but we can take a look at it with higher magnification to try to identify any other features that could confirm this. So again, here we go. Alright, so epithelium here is starting to look a little bit more columnar. So simple columnar epithelium. Just decrease the contrast a bit to help us visualize things a bit better. Okay, I don't see any evidence of glands, and there's definitely no evidence of any kind of cartilage. fairly small lumen, and we can just see basically a little bit of smooth muscle here. So there's the muscularis mucosa. This is the lamina propria, and out here is the tunica submucosa. Okay, so that would be, again, a fairly small bronchiole. And again, the material that's kind of eosinophilic inside is likely to be some of that clara cell protein that's kind of like the Teflon that's preventing these cells from sticking to one another if this collapses in. Again, without the cartilage plates, there's really not much preventing this structure from just kind of flattening it against itself when there's negative pressure inside of this structure. Let's go back out, see if there's anything else I can find in here that's helpful to us. Okay. Okay, so let's take a look at some of these alveoli. Okay, so let's zoom in to 100x. So we're going to take a look at some of these very, very thin walls here. Go to 400x. And I want you to be aware of just how thin these things really are, because what you're looking at are walls of two separate alveoli. There's a lumen of alveolus 1, lumen of alveolus number 2. So that means that there's an epithelial cell at the surface on this side. There's also an epithelial cell on the surface on this side as well. And in between them, there's still a capillary. Okay, so there's going to be an epithelium surrounding that capillary as well, an endothelium. And again, you're going to have some basement membranes in there as well. So there's a lot going on here. Okay, let me see if I can find an interesting region here. Looks like there's a capillary visible in here, so I'll try to zoom in on that part and see if we can get a good look at this area here. Okay, so this is the wall of one of the alveoli, or I guess a couple of alveoli, because there's an alveolus on either side. So where the pointer is the lumen of one. Here's the other one. And so what you're seeing here is a capillary. So there's a red blood cell visible here. Maybe bump up the contrast a little bit. Okay. And again, on either side of the capillary, you're going to have type 1 alveolar cells or pneumocytes that are spread as thin as possible to allow for very easy and very efficient gas exchange. Okay, so we're going to probably just stop here and try a different slide. Thank you.